Наследственное вещество имеет уникальное свойство – создавать точную копию самого себя, то есть удваиваться. Это происходит в период между делениями, в интерфазе. Хроматиновые нити настолько плотно упакованы, что в ядре умещаются молекулы ДНК общей длиной более метра. С началом деления, митоза, хромосомы спирализуются, становятся толще, короче, ядерная мембрана исчезает, а центриоли расходятся к полюсам, формируя двухполюсное веретено. Хромосомы выстраиваются вдоль экватора. Теперь каждая хромосома состоит из двух хроматид. И хроматиды расходятся, подтягиваясь к полюсам. Дочерние клетки получают одинаковый набор хромосом, следовательно, и генов, определяющих признаки будущего организма. Каждый организм имеет постоянный набор хромосом – кариотип. Это непременное свойство всех живых существ. Кариотипы отличаются по числу хромосом, их размерам и форме. Именно индивидуальность кариотипа сохраняет видовое постоянство из поколения в поколение. От курицы – цыпленок, от человека – человек. Кариотип человека всегда состоит из 46 хромосом, 23 пар, полученных от отца и матери поровну.
В жизненном цикле клетки выделяют два этапа: интерфазу, в результате которой клетка готовится к делению, и митоз, при котором из исходной родительской клетки образуются две дочерние клетки с таким же набором хромосом. Интерфаза в клеточном цикле занимает самый большой промежуток времени. Идет активный рост клетки, синтезируются белки, запасается энергия за счет синтеза молекул АТФ. В этот период отчетливо видны ядро и ядрышко. Хромосом не видно, они раскручены и равномерно распределены в виде рыхлой массы по всему ядру. В интерфазу происходит удвоение хромосом. В основе этого процесса лежит процесс удвоения или репликации ДНК. Удвоенная хромосома состоит из двух половинок, каждая половинка – сестринская хроматида, содержит по одной двухцепочечной молекуле ДНК, соединенной с хромосомными белками. Хроматиды полностью идентичны друг другу, соединены центромерой и тесно сближены друг с другом. Удвоенные хромосомы по-прежнему имеют вид тонких нитей в виде клубка, занимающих весь объем ядра клетки. На втором этапе клеточного цикла происходит митоз. Это непрерывный процесс, который для удобства разделяют на 4 стадии. Первая фаза митоза – профаза. Наибольшее значение в этот период имеет скручивание хромосом. В результате они преобразуются в компактные структуры. Одновременно происходят изменения и других клеточных структур. Исчезает ядерная оболочка и ядрышко, из микротрубочек формируется веретено деления. Во вторую фазу митоза, метафазу, хромосомы лучше всего видны в световой микроскоп. В начале этого периода хромосомы лежат непосредственно в цитоплазме. Далее к центромерам хромосом с двух сторон прикрепляются нити веретена деления, и хромосомы начинают двигаться, пока центриоль не окажется на одинаковом расстоянии от двух полюсов. В результате хромосомы выстраиваются на экваторе клетки. В анафазе, в третьем периоде митоза, центромеры хромосом разделяются. сестринские хроматиды становятся самостоятельными хромосомами и с помощью нитей веретена деления начинают передвигаться к разным полюсам клетки. Таким образом, к каждому полюсу отходит равный набор хромосом. В последнюю фазу митоза, телофазу, хромосомы на каждом полюсе раскручиваются и принимают вид тонких нитей. Вокруг них формируется ядерная оболочка и появляется ядрышко. Далее происходит распределение клеточных органоидов, и все завершается делением цитоплазмы. В результате клетки разделяются на две дочерние клетки, полностью идентичные материнской. Митоз имеет универсальный характер. Он протекает сходным образом у всех видов, клетки которых имеют ядро. Митоз обеспечивает равномерное распределение наследственного материала, благодаря чему поддерживается постоянное число хромосом, обеспечивается рост организма, а также сходство потомства с родителями из поколения в поколение при вегетативном размножении.
An adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into three distinct phases. During G1 or gap1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as two sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach, by proteins in their centromeres called kinetochores, to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all three phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency of the checkpoints during interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves. This process allows for growth and repair to prolong overall physiology as well as life itself.
Cell division is required for an organism to grow, mature, and maintain tissues. During the mitotic phase, a cell will undergo mitosis to form two new nuclei, and then divide to form two new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into two new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the two sets of chromosomes. Mitosis is now complete. After mitosis, two new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of three stages called G1, S, and G2. G1, or gap 1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap 2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and 2 centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 80 minutes. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process. At the same time, protein strands called microtubules appear from the centrosomes in animals. Finally, a structure found within the nucleus, the nucleolus, disappears. Next, prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands or spindle fibers are growing from the centrosomes. These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell. Now the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart, causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the 2 ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of 2 cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the 2 halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the 2 new cells are now fully in the G1 stage of interphase and ready again to begin their growth. Let's watch the process one more time. Mitosis begins with prophase. Notice the DNA condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear membrane breaks down. Metaphase occurs when the chromosomes are aligned at the center of the cell. During anaphase, the chromosomes are moving apart. The telophase stage is marked by the appearance of new nuclear membranes. This is the end of mitosis. Finally, the splitting of the cell occurs during cytokinesis. The 2 new cells are now ready to grow and perform their specialized functions.
Mitosis. In this module, you will learn about mitosis. Mitosis is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into 2 identical daughter nuclei. It is divided into 4 key phases: prophase, metaphase, anaphase, and telophase. An additional phase called cytokinesis occurs after the complete nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest. This period is called interphase. In interphase, the genetic material of a cell increases. Prophase. During prophase, The chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has two sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores and the daughter chromosomes, along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into two sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to decoil. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage furrow appears in the plasma membrane, which deepens and divides the cytoplasm into two daughter cells, each containing a nucleus. In plants, new cell wall formation is triggered at the center of the cell by the formation of a cell plate. The cell plate grows outwards till it meets the existing wall, resulting in two daughter cells. In this module, you have learned mitosis is the process of separation of nuclear chromosomes into two identical daughter nuclei. Mitosis involves four key phases: prophase, metaphase, anaphase, and telophase. In prophase, the chromatin condenses and becomes short and thick to form the chromosomes. In metaphase, microtubules of the spindle interact with the chromosomes. In anaphase, the sister chromatids separate out at the centromere and move towards the opposite poles as the microtubule becomes shorter. In telophase, daughter chromosomes arrive at the pole and chromatin starts to decoil, followed by the reformation of nucleoli and nuclei.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug-of-war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.